Well, all right. I'll make a few introductory remarks to start with as our participants continue coming in. I'm Evan Siegel, the CEO, President of Ground Zero Pharmaceuticals, a regulatory affairs and product development consulting firm. This is the next in a series of webinars that discuss early medical product development, early into medium level medical product development. This one was particularly oriented towards Aussie firms and sponsors of such products. We have a large presence in Australia, have been there since 2003 with clients, and incorporated a subsidiary in 2006. I'm an adjunct professor at the University of Queensland, so have a lot of continuing connection with your lovely country. And we have quite a few clients down under. So I'm very honored to be able to give this webinar set up at a time where all of you could participate without getting up frightfully early in the morning. The title of this one tonight is CTN to IND, Maximizing Your Initial Data to Move to IND and Attract Investors. And many of the actual precepts here would really hold for invasive implantable therapeutic medical devices, as well as IVDs to some extent, and companion diagnostics. But this is primarily oriented towards small molecule, biologic, and complex biologic therapies. The first concept is product development, a regulatory perspective. We believe that regulatory affairs is really the center of all medical product development. And the reason for that is no matter how good your chemistry manufacturing controls information may be, or your early clinical data, if you have such data, and your non-clinical testing, pharmacodynamic data, PK, ADME, et cetera, without the regulatory process, the program goes nowhere. So you need to integrate your various technical data into a regulatory environment, regardless of that environment. It might be TGA, might be FDA, it might be EMA, might be Health Canada, or might be the Japanese medical authorities. You still need to use regulatory affairs as the medium to integrate your data and present those data in a favorable manner to the regulatory authorities. The big question will come up, well, why should Australian researchers engage with the FDA? Since 2003, we've noticed that more Aussie companies are willing to engage earlier. At one time, in general, Aussie companies were allergic to going to the FDA for fear they might get a negative answer for questions they might ask or the program in general. And they've realized that as very, very tough regulatory authority, if you pass the FDA test, you actually will have much more interest from financiers, particularly outside of Australia, within Australia as well. And in general, your program can get a more favorable launch across the globe. So going to the FDA early is really an important concept to take away from this. When we think of 4 cardinal questions from investors who are actually going to be supporting most of your initial programs. Do you own it? Is the IP and patent status favorable? What about marketing exclusivity, depending on the regulatory pathway you take and in whatever region you place your product? Does it work? No matter how good the concept, no matter how wonderful the number of papers that have been generated with your IP, if you can't show preclinical and non-clinical efficacy based on pharmacodynamic principles and early human clinical data, you really don't have a program that's terribly viable moving forward. So does it work is a very important concept for investors. Can you get it approved or licensed? In fact, a product may actually be able to get approved, but may not sell well. A clear example of that is the several inhaled insulins, which cost collectively more than $2 billion to develop and never sold well. And the reason for it is because that inhaled insulin method of introduction of insulin was very upsetting to many insulin takers, did not allow for stringent dose titration in many cases, and depended on the particular characteristics of the nasal mucosa in individual patients, any concomitant disease they might have had, medications, et cetera. So essentially, getting it approved is obviously important to investors and to others. The regulatory status and prospects of the product through the regulatory process, and any special status, orphan drug status, which conveys 7 years of marketing exclusivity after approval in the US, 10 years in the EU, and varying degrees in other regions. The final question is, can you market it? What are the competitors like? What is the acceptability to healthcare providers and patients? And really that discussion of inhaled insulin is important on the marketing front as well. The biggest companies have failed with some product launches simply because the product was not easily marketable. So going into, do you own it? There we have the IP patent status, marketing exclusivity mentioned earlier. Rare pediatric designation is very important these days. Additional indications. What's the prospect for your product to have additional indications added after your marketing application is approved? Are you a small molecule versus biologic? A small molecule first indication in the US typically gets 5 years of marketing exclusivity. A biologic gets 12 years. So even though biologics generally take longer to develop and to get approved than small molecule drugs on average historically, the biologics have a great deal more marketing exclusivity, at least in the US. And the timing of market entry. What are your competitors like? What is the best time to market a product, given economic conditions or price controls or some other reasons? And all of these considerations are very important for investors and should be important to the management of the company and its board. And jurisdictional differences. Obviously, patents are not universal across the world, so one must consider the various regions and the kind of patent protection and challenges that occur in these regions. Does it work? The quantity versus quality of data, lab notebooks versus reviewable reports. You go to a sophisticated investor and say, we have lab notebooks full of great data. They'll turn around and say, show us the reports generated therefrom, and we'll talk to you about financing you. In vitro versus in vivo non-clinical data. In vitro data are cheap and quick to obtain, but they don't hold the amount of impact that in vivo data hold in an animal model of disease for the vast majority of drug and biologic products. The selection of animal models, are they relevant or irrelevant? It's been said that anything will cure a mouse with a tumor on it. And to some extent, that's very true. Most mouse model data sets are very positive, but the conveyance of that result to the human situation may be very difficult. The number of animals, doses administered, controls. A sophisticated investor will look at these kinds of issues. Human data. Do you have human data? If you do, are they obtained via an open, small, anecdotal study or in a controlled study? Controlled data always have much more impact than anecdotal data to all regulators and to investors as well. You may say, well, in serious or life-threatening disease, you can't do a controlled study. Well, actually, you can. You generally don't do a placebo-controlled study in oncology, but you could do standard of care versus standard of care plus your experimental agent. And if you show significant incremental differences, then you may really have something there. The choice of comparators is very important. And the relevancy of endpoints selected. All of this fits under the moniker of does it work? What kind of data support do you have for the product? Then can you get it approved? We've talked about this already, the regulatory status. Have you had a pre-IND consultation with the FDA? Do you have an IND in place? What is your IMPD and CTA status in Europe? What about a CTN in Australia? And if you have no clinical data, that's fine. You then have to design your program for a first-in-human study, which can often be done efficiently, relatively cheaply in Australia. The magnitude of effect is very important. You don't just look to statistical significance in your data, but you also look to the magnitude of effect for clinical relevancy. And the regulatory agencies and the investors really look at this. If you have a large enough study, by chance, you may get a statistically significant effect, but if the magnitude of clinical effect is really very small, the viability of the program moving forward may be somewhat in question until you generate more data. So early versus later studies are important. Single versus multiple indications. Do you have a receptor targeting monoclonal that can be used to treat more than one indication? That's very important in terms of the value of the IP, the value of the data, the value of your program as a whole. Special status and designations we mentioned earlier. And you need to establish timelines, costs, and realistic milestones. I'm going to emphasize realistic because most of our Aussie clients have unrealistic milestones established by their investors and their boards. And biology cannot be rushed. You can do everything possible to bring in good people, to bring in good consultants, to work with good investigators, to work with good CROs, to work with good consulting groups. But there are a certain number of exigencies that exist when it comes to generation of biological data. So realistic milestones need to be in place. Otherwise, you'll always be playing catch up with promises you've made and promises you have not kept. Can you market it? We mentioned it earlier. The competitive arenas can be indication specific, platform specific. Thought leaders, key opinion leaders are important. Patient compliance, very important. Is your product very easy for patients to take, with which to comply? Manufacturing, shipping, storage, and delivery seem mundane, but they're actually very important. CAR T cells are a very sensitive area. The storage can be very expensive. The stability can be limited. The shipping can be difficult if in fact you have a central repository. If you don't have a central repository and you're creating the CAR T cells in situ, then you have to be very careful about consistency for treating patients for a particular indication. So all of these factors need to be looked at when your program is before investigators. Investigators and investors. Region-specific issues such as cost. As we all know, the US is outrageous in cost of drugs. It also is a great market for those who wish to market their high-tech products. So there's an issue there in terms of public acceptability versus cold, hard recovery of your investment dollar, the investors making their money, and the issue of medical practice, all coming together in those region-specific areas. The ease of conducting clinical trials. Where do you go? Australia has a relatively low bar to conducting good quality clinical trials in terms of regulatory oversight. The regulatory oversight is typically through the ethics committees and not through TGA for the vast majority of all clinical trials. In the US, they all go through the FDA. So there's a big difference. Risk benefit for the program. Well, obviously, if your program is very risky from a marketing perspective, even if it shows a great deal of efficacy and acceptable safety, the risk of putting marketing dollar behind the development dollar also comes into play. Well, another key thought, let the data speak and avoid unrealistic projections. You've already seen some of this as a theme. Unrealistic projections kill companies. Realistic projections protect companies and give them credibility. And credibility attracts investment dollar and also is a very powerful, positive force in the regulated environment. If you're realistic and if you're honest and honorable with the regulators, you'll likely get a good break from them when it comes to a decision which could go either way. And the data will always speak. You can talk up the theory, you can talk up the concept all you want. You can say we've got a Nobel Prize winning concept here, but I can tell you the history of Nobel Prize winning concepts that led to effective drugs and biologics is actually very small. It's accelerated somewhat, but it takes a very long time for a Nobel Prize winning concept to lead to a saleable drug and biologic product. So the data themselves from your product, from your project, are what will speak best to investors and regulators. So INDs versus CTNs. What do you need for clinical trial initiation? Well, the CTN, as I'm sure most of you know, is a notification to the TGA, not a full dossier like an IND. The ethics committee takes responsibility for primary review of documents and the good clinical practices conduct of the clinical trial. You basically need the title, brief details of the trial, the site and the investigator, plus a certification by the latter and by the ethics committee that's applied in the CTN. And the ethics committee reviews the protocol, reviews the investigator brochure, and typically summaries of toxicology and pharmacology to assess the appropriateness of conducting the clinical trial. One key issue here is that a CTA in Europe, a CTN in Australia, are not a dossier for the product. They're individual clinical trials that are supported by information from the dossier. An IND is a complete dossier. You could conduct 20 clinical trials under an IND. Once you have the IND open, you just need to submit new clinical trial protocols to that IND, as long as it's for the same product and within the same FDA reviewing division. So you could have 2 or 3 oncology indications that are all reviewed by Division of Oncology 1, one IND for that product, and be looking at 3 or 4 or 5 indications with 5 or 6 or 7 or 8 or 10 clinical protocols. So there's a difference in that clinical trial specific dossier versus the product dossier, which begins with the IND. The CTN also contains the ethics committee approval letter, brief information on the experimental product, the name and description, and that toxicology, pharmacology expert assessment is typically included. And that's done by an expert tox pharm person who takes a look at the extant data that support the product, whether it's a first in human product, a phase 2 or a phase 3 product, and make an assessment and are willing to attest to the fact that the data, the extant data, support that clinical trial. An IND is typically submitted modularly now. The IND regulations, if you look them up, are very old now, and they contain 11 or 12 or 13 items, they're called. In reality, all commercial INDs need to be submitted in eCTD format. The CTD is a common technical document that's good in Europe, Japan, the US, China, many other places. And this is what it looks like. The region-specific administrative FDA forms and certifications for an IND, the investigator brochure, and the drug label in Module 1. Module 2 are the summaries of quality, safety, and efficacy. Module 3 is the complete quality dossier, and this is complete. It's basically a summary and detailed reports on all of the CMC data. Module 4 is all of the CMC, all of the non-clinical study reports, pharmacodynamics, toxicology, safety pharmacology, ADME, PK. And Module 5 is any clinical study reports. If it's a first in human trial, then Module 5 is typically going to be empty or might contain the clinical protocol that you intend to support with the IND for a first in human. The chemistry manufacturing controls or product quality, basically for a phase 1/2 IND, and I use this because a lot of the work that's being done in Australia and that we do is for serious or life-threatening illness. Often your first in human will be in patients, not not human volunteers. So you might call it a phase 1/2. And the typical 1/2 IND package contains the GMP or GMP-like clinical trial material documentation. For US, you do not need full GMP clinical trial material for a phase 1 or a phase 1/2 clinical trial. For phase 2 and 3 and beyond, you must have full GMP. Then you have things like the characterization, analytical data, strength and potency, stability data sufficient to cover your dosing for that first clinical trial, the drug substance, drug product data, and all of the rest. And by the way, you will be able to obtain all of these slides from Lauren Magis after this seminar. So you don't have to worry about taking extensive notes or me covering every single word. The preclinical studies for an IND can be GLP and non-GLP, but all of the toxicology and safety pharmacology studies and any others that are intended to provide primary safety for that clinical trial must be GLP compliant. And so the non-GLP might be things like an ADME study or a mechanistic study or a pharmacology study that is not related to safety, but is related to mechanism of action or showing that you have some signal of efficacy. So those don't need to be GLP compliant, but the primary safety support studies must be GLP compliant. And if there are studies that employ specialized test systems which cannot fully comply with GLP, the FDA allows that there are some very unusual rarefied receptor type studies and localization studies which cannot be compliant fully with GLP, and those can be submitted to the FDA and will be accepted. But you have to identify the areas of non-compliance and evaluate their significant relative to the overall non-clinical package in the IND. The typical early IND package includes, as I mentioned, the pharmacodynamic studies, in vitro and in vivo animal models, the GLP tox studies in 1 or 2 species. Now you're going to say, well, the guidelines talk about 2 species. Yes, but for advanced cancer and frankly, other serious life-threatening illness, you can often not provide 2 species. You need a rodent or a non-rodent, but you don't need both in certain circumstances. And in fact, if you only have 1 relevant animal model, which bears the possibility of predictability of safety in humans, then you only use that species. You don't waste animals' time, money, and resources with an unnecessary and irrelevant study. The genotoxicity panel is standard for small molecules. It is often not for biologics. And the safety pharmacology panel is required for biologics and small molecules, but that can be combined often with toxicology studies. Safety pharmacology panels are the CNS, the respiratory, and the cardiovascular single dose studies, which look at those specific systems in animal models and attempt to examine the effect of a particular experimental agent on those systems, in addition to the general toxicology studies, which cover many of the body systems at the same time, and ADME and PK information. You want to know where the product goes when it's introduced into the body, and the first means of determining that is in a relevant animal model. You can enhance the potential for success by generating only high quality data, forego large numbers of exploratory studies for publication in favor of reviewable reports. I've been saying this for 17 years. The university generates a huge amount of wonderful intellectual property with great experimenters, great investigators. But their raison d'etre is often submitting papers for publication. Papers for publication do not substitute for reports of pharmacology, toxicology, safety pharmacology, et cetera, studies. So if you're going to develop a drug or biological product, you need to rein in those who would generate dozens of papers showing how wonderful the concept is, how great the animal data are. But those studies in publication are literally only supportive. They are not primary for the regulatory agencies. So you really need to be careful how many publications are generated compared to how many reviewable reports are generated. And I mentioned earlier, consider testing only in relevant species, avoid ancillary seeding studies where you're kind of throwing out a little bit of data and saying, wow, it works. One patient was stable and a few of them progressed and a couple of them died, but one of them was stable. That's not a good data set. That's anecdotal and it involves 1 or 2 patients. So seeding the product across many indications in 1 or 2 or 5 or 10 patients really doesn't get you a good reviewable data set. It's much better to look at your mechanism, look at your early animal data, choose 1 or 2 indications, follow them up with backups, and generate a good body of data in 10 or 15 or 20 patients that means something to the regulators instead of a few anecdotal patients here or there. And if you want to say, well, the cost is prohibitive, well, it's much more expensive seeding 5 useless studies than 1 or 2 good studies. And that's what we found over 21 years of company history, looking at hundreds and hundreds of data sets. It's better to do a small number of definitive studies than to do a whole host of small seeding studies that really get you very little in the form of regulatory interest, or for that matter, investor interest. For serious or life-threatening disease, there are facilitated and accelerated approaches. And I'll go into some of those, but you can always ask questions about those. There are programs such as Fast Track, Breakthrough Therapy Designation, the orphan drug mentioned earlier, Rare Pediatric Disease Designation, and other special programs, which do give you an edge, but your product and your program and your data have to satisfy the requirements for obtaining those special indications. So what are critical necessary reports from early data? Primary pharmacodynamics. You have a target. How does the product interact with that target? How does it work in an animal model of disease where that target exists? Secondary pharmacodynamics. What about off-target effects? Off-target effects can have 2 outcomes. One is that you can have a fortuitous effect on another pathway or another target, which might give you greater efficacy. The other off-target effect is you can have pharmacodynamic toxicity because of off-target effect that does nothing for your primary efficacy. So the ratio of on and off-target effects is important in the reports that you generate and submit to regulatory authorities. The safety pharmacology, the toxicology, the ADME, and the bioanalytical methods. If you have all of these and nothing more in the non-clinical arena, you'll probably be in very good shape to at least go to the FDA for a pre-IND meeting. A very quick example, a pharmacology report from early laboratory data. These are the types of studies that are often published, but not provided to investors, consultants, and others in reviewable reports. A pharmacology report, a prospectively written protocol, all the amendments, a detailed methodology, detailed description of a study design. What animals did you use? What was the test system? What were the dose levels? If you have 1 dose, it's a dead report. It's worthless for drug and biologic development. If you have 2 doses, it's generally worthless. 5 doses is more like talking about it, because then you can profile the pharmacodynamic activity and look for dose ranging. What about male and female? If the disease exists in males and females, you only test males or females, it's a semi-worthless report because you don't know what happens in the other sex. The dose levels used, detailed procedures for the administration of the test article and the collection of study protocol parameters, and the results of all parameters evaluated along with analysis and interpretation of study data. That's a report. Every study you do to provide support for a new drug or biologic should have a report like this when it comes to pharmacology, pharmacodynamics. Well, we'll shift gears a little bit and talk about a key meeting with the FDA, a pre-IND meeting. You may submit an IND directly to the FDA with data from a CTN or other sources or both. But the pre-IND meeting is a very important meeting that allows you to have an early discussion with the agency before you submit your expensive to generate IND. The role in strategic development can be to identify studies that will support the initiation of clinical trials, which de-risks the IND submission, resolves potential safety issues before you even get to the IND stage, and discuss available methods to enhance your development. For example, the orphan drug, fast track, breakthrough, animal efficacy, rare pediatric disease designation, regenerative medicine, advanced therapy designation. Not all products will be subject to these, and the vast majority of products will not be able to get all of these. And really, you don't need all of these, but each time you get a special designation, it can be very helpful. An anecdotal example is a company I'm sure you all know well in Australia that just announced a rare pediatric disease designation, orphan drug, and fast track for their product, all within a 2-week period. And we know them well, and you know them well. And that about tripled the stock value, just getting those special designations. So it's something where you might want to consider that kind of a discussion with the FDA early on, which will help give you a roadmap towards your development profile of the product. The pre-IND meeting package contains background information, including your development strategy, detailed information on your drug substance drug product. When I say detailed, I don't mean what's in an IND or a marketing application, but a good summary, including a summary manufacturing process diagram for the drug substance drug product, other information on the CMC, summaries of the available non-clinical data, just summaries, brief description of the proposed first IND clinical protocol. You generally don't need a protocol, but a protocol synopsis is fine. If you're in advanced phase 2 or phase 3, we suggest that you do submit a protocol to the pre-IND, but that's your choice. And then specific questions to be discussed at the meeting. You don't put your program before the FDA and say, what do you think? You say, in the CMC, we've got 2 months of stability. We believe that this will cover the 1-month duration of the first clinical trial proposed in the IND. Do you agree? Or we have found only one relevant species. It's the dog. We've done safety pharmacology in the dog as well. Here are the summary of results in the dog. We feel, given the fact that this is an advanced cancer indication, this is necessary and sufficient to move ahead into the IND. Do you agree? That's the way you cast questions to the FDA in the pre-IND meeting. And the meeting will give you written minutes from the FDA, whether it's a written response only, a telecon, or an in-person meeting. I can tell you right now, the number of in-person meetings is 0 for multiple reasons. But a telecon meeting can be very valuable. A written response only meeting gives you a full FDA assessment of your program and the questions you've asked. However, you don't get the interactive immediate interaction with the FDA. So we normally suggest you ask for a telecon meeting, and that way you get the best of both worlds. A modern regulatory strategy package, realistic review your product indications, assess the emerging safety and efficacy signals. All programs are going to have safety problems. All programs are going to have some efficacy blurps along the way. So how to assess and handle those realistically, how to deal with the fallout, how to perhaps enhance the program after you do see an adverse safety signal or efficacy signal is important. So too, if you see an unexpected positive safety profile beyond what the animal data showed, and or you see an incredible efficacy signal, then you might be able to accelerate your program, and you can use the special designations, one or more of them, to do that. We suggest meeting often and early with regulators, and also consider new clinical approaches. Phase 1 expansion cohorts is done all the time, particularly in serious or life-threatening illness. Multiphasic protocols, instead of a phase 1A, phase 1B, phase 2A, phase 2B, phase 3A, phase 3B, et cetera, we suggest a phase 1/2, 2/3. And if your program is suitable for the same, then you can get to yes with far fewer clinical trials and a much smoother development program that doesn't involve the stopping of your clinical trials for 3 months or 6 months or longer while you're evaluating your previous clinical. Adaptive designs, targets and markers across tumor types and tissue of origin in oncology. This is emerging and very, very important. It used to be that if you had a brain tumor or you had a lung tumor or you had a soft tissue sarcoma of the arm, those were considered very discrete diseases. Now we know that, that certain types of breast cancer have a great deal in common with certain gastrointestinal stromal tumors in terms of the targets, in terms of how the tumors begin and grow. And you have agents which can act against what used to be considered diversely different disease entities, but in fact have the same type of histology and the same type of responsiveness. So you can expand your investigations and your indications with your same agent by looking across tumor types as long as they have verifiably the same markers that enable binding of targeted agents, immunotherapies, etc. Fast Track designation is one of those special programs. You have to have serious illness. The designation is given to a specific indication and a specific product. You have to demonstrate an unmet medical need, some preclinical or early clinical promise based on data that you have generated. The request for the designation is a formal submission to the FDA, typically no more than 50 pages. You have to allow for a 60-day review window, and if the designation is not granted, you can reapply. And the FDA will always now, these days, in a turn-down letter, will provide suggestions on how you might enhance your program. So you get somewhat of a roadmap to coming back and requesting the designation again. You can request Fast Track designation anytime up to the submission of a marketing application, but the earlier you get it, the more special help you get from the FDA, additional meetings, additional consultations, because they've identified your program as priority. Breakthrough therapy designation is like Fast Track on steroids. The molecule can be alone or in combination. Again, serious illness. You have to make a clinical comparison with US approved therapies. That is a drug, a biologic drug, a medical device, or what have you that is available through sale in the US, and show substantial improvement over all existing therapies. So the breakthrough therapy designation hurdle is quite high. However, if you get it, it gives you accelerated review, accelerated meetings, kind of, again, breakthrough, as a fast track on steroids. And the typical breakthrough therapy is approved in 3 to 4 months after submission of a marketing application. It's a very accelerated program, and the FDA will grant specific meetings to expedite development. They will just review every single clinical trial for design to look for efficacy. So essentially, you get your own little reviewing group for your breakthrough therapy. How do regulators and investors review clinical trial protocols and strategies? Well, you want to design a clinical trial that can carry a punch with both regulators and investors. Some housekeeping: you should follow the latest guidelines on clinical trial protocols, include full procedural information on the trial, include descriptive statistics, and an analytical plan. Those are fine for phase 1-2. Again, this talk is oriented more towards early programs. The informed consent form should be included. It is actually not required to submit the informed consent in an IND. The FDA has asked for informed consent in every IND a few years ago that we did not include. So now we include an informed consent every time an IND is submitted. Include DSMB information as applicable. That's a safety review committee, a safety monitoring committee, a data safety monitoring board, whatever you want to call it. It's pretty much the same concept. And you do not need exhaustive references in a protocol. The more references you include, the more references you'll need to include in an IND. And the FDA reviews those. And there's no sense cramming up reviewers' time with unnecessary references. So you really want to keep the reference list small for both protocols and INDs. Well, the concordance of the protocol with other information thus is important. The protocol should be appropriate for the stage of investigation. I would certainly avoid a 400 patient oncology protocol in phase 2 or phase 1/2. It's not going to fly. The safety support for every protocol is critical. The pharmacology data should support the proposed study. And you do not need to prove the mechanism of action in the US. There are other regions where mechanism of action is really important, such as the EMA region. But you really, I mean, mode of action is helpful. It can explicate what you're doing, why, how and why the product acts, and perhaps enable you to predict some off-target effects, pro and con, but you don't need to prove a mechanism of action to get a protocol or an IND through the FDA. And manufacturing data should allow for thorough review of the process and quality attributes of the drug substance and drug product. So when you submit a protocol, and it's a fairly aggressive protocol, your CMC data in the IND should be up to the protocol. So as you go from phase 1 or phase 1/2 to phase 2 to phase 3, the CMC requirements toughen up at the FDA. You need to have much more validation, much more analytical methodology certainty, much more advanced methods for assessing purity, potency, structure in a biologic, et cetera. So you're basically working in lockstep. As I said at the very beginning, you want your non-clinical, your clinical, your CMC, and your regulatory technical areas to be in harmony with one another as you move through the development program. Well, there are some special issues for biologic and biotechnological programs. The group at the FDA that reviews biological, biotechnological products is both CBER and CDER. CBER regulates cellular products, products composed of human, bacterial, animal cells, gene therapy products, vaccines, antitoxin, blood and blood components. CDER, Center for Drug Evaluation Research, actually evaluates small peptides and monoclonal antibodies. So it used to be that CBER had all of the biologics, and that's changed substantially since 2003, 2004. So you need to know where you're going to be placed. Some biological, biotechnological investigational products warrant additional containment considerations: pathogenic microorganisms, spore-forming microorganisms, live viral vaccines, gene therapy vectors. All of these are active programs that we have and others have before the FDA right now. So, you know, one size does not fit all. That's probably the message for this. And you need to very carefully consider the kind of regulatory scrutiny you will get with a biological or biotechnological product. It's difficult to predict the impact of changes in quality attributes on safety. If you change one amino acid in the sequence of a biological product, you may change the safety profile. And phase 1 studies are where there's a special emphasis by the FDA. What you put before the FDA in phase 1 needs to be carefully construed, and you need not to have a lot of variability in your analytical data for that early clinical trial material, because without the human clinical data to see what happens with your product and to look at its purity and potency effects in the human, you need to be very, very careful in that early characterization. You can't have very impure biologic going into humans in phase 1. Reproducibility must be established for control and documentation of the production process. And you should retain samples to establish batch-to-batch reproducibility. This is especially true, again, for biotechnologically derived products, say those obtained through recombinant means, because your entire development process for your product is a continuum to the FDA. It's not as if you had a few phases and the old data are the old data and the new data are the new data and they never connect. You need a really solid developmental profile for your biotechnological product, even in your IND, and certainly in your marketing application, which would be a BLA, Biologics License Application. You need the provenance and history. You need to have master cell banks and working cell banks. You need to be very, very careful as compared with a synthesized small molecule, which is very different than a biological. And with that, I'll let you ponder a bit and put some questions up. If we have any, then I'll take a look at the chat if I can get it. There we go. Okay, first question is, for IND submission, can a GMP-like product be used for GLP toxicology studies? And the answer is resoundingly yes. As a matter of fact, if you don't intend to have full GMP at phase 1 or phase 1/2, it's preferable that you use the same material you intend for the first human clinical in your GLP tox studies, because then you have 100% concordance. You don't have to supply a table to the FDA showing the characteristics of one against the other. So the test article and the clinical trial material are one and the same. Another question, does the drug need to be GMP to enter first in human in phase 1? The answer is no. It can be GMP-like in the vast majority of cases. The FDA has a guidance on this, which is quite old, but has recently been recapitulated. Unlike most other regulatory authorities, you don't need full GMP to enter phase 1, or we've extended that to phase 1/2. And GZP has never put a phase 1/2, say first in human, in patients. We have never had it rejected because the material wasn't full GMP. GMP-like with good characterization and reasonable specifications of purity, potency, structural integrity, et cetera, have been accepted. Let me, before the next question comes in, say that I really want to thank LSQ and Sam South of WA, Lauren Mages of our own GZP, and others who contributed to setting this presentation up and setting up the publicity and publicizing it. I appreciate it very, very much. So any questions you want to ask, I'm here for pretty much as long as you would like. Okay, next question. In the case of using viral vectors, does your GMP batch, I guess it means should your GMP batch be from the same cell passage number as that of the GLP batch? I would say that it's preferable, but as long as you have concordance and you've tested what passage does to your product, again, it's product quality attributes, I would say no, it does not have to be from the same passage number. It should be reasonably close, but I don't believe it needs to be the same. Any specific guidelines for combining device drug regulation? Yes, there are a whole series, and actually I was the editor of one of the seminal books a few years ago on drug device, device biologic, and drug biologic combination products. So there are plenty of guidances on the combination products website, sub-website at the FDA. Yeah, and you do have to, it's a bit draconian. If you have, for example, a drug device, you need to follow GMPs for both the drug and the device. That's the ruling by the FDA. So you have to be very careful about your quality system or else the quality system of the CMOs that you use, and make sure that you audit them and make sure that everything's concordant. Next question, can a product which is analyzed using developed methods but not validated be used in GLP toxicology studies for IND submission? Yeah, if the methods are qualified early on, they don't need to be validated, but they should be validated later in the program. And yes, they may be used in GLP tox, but I would use the same method for the clinical trial material that you use for the GLP tox test article. Or if you're talking about, yeah, that's product analysis. So I correctly interpreted that question. The slide deck, yes. If anyone wants the slide deck, please, or a call with Lauren or me, feel free to do that, to set that up anytime after this meeting. Okay, question in oncology, is it possible the FDA would one species tox to open the IND with a commitment to complete second species during phase 1? Generally, the FDA says you either need 1 or 2. Making a commitment if 2 species are really considered necessary is not generally going to work. But if a study is underway and you have an interim analysis, for example, that can certainly help you qualify that second study. In oncology, in advanced oncology, you actually rarely need 2 species. Our experience has been resoundingly that you don't need 2 species. It helps to have 2 species, but it's not required. The S9 with revisions guidance on development of oncology drugs and biologics specifically talks about one relevant species as opposed to 2. But I suppose if you're planning on doing 2 and you want to submit early with the 1, you could just say that you'll submit. There's a little danger in that the FDA will say, well, why don't you just wait for the results of the second study? So it's a kind of a judgment call. Is ADME required to submit for CTN? That's an excellent question. Rarely asked that. What you need to submit is what the ethics committee requires for that specific situation. ADME is quite important because if you don't know the fate of the product, you really have no idea of your dosing or dosing interval. So at least some PK data would be, I think, important to submit for a CTN. Must advanced disease include METs? What about TNBC where there are a few therapies? I'm not quite sure I understand. Do you mean, by METs, do you mean metastatic lesions? I mean, advanced disease doesn't have to have metastatic disease. It can simply be a very large tumor which has spread locally. That's still considered advanced disease for some tumor types, and depends on whether the organ or organs involved are grossly affected in function. If they are, you would call that advanced disease. So, I mean, you can call an advanced disease without METs. TNBC, where there are a few therapies, well, if you have an unmet medical need, although TNBC is being studied extensively as we speak, if you have something which appears to be very effective in TNBC model or early studies, then I would say that would have a priority for your program, if that helps. Yeah, metastatic lesions, yes. Yeah, I think I just answered that. I mean, advanced disease doesn't automatically mean metastatic lesions. Well, great questions. Please keep them coming. This is fun. Okay, how does the FDA view data obtained in Australia under CTN, i.e. not under IND? Well, as you know, well, as you may not know, as you know, you can do a CTN study in Australia under an IND. Our particular preference for our Aussie clients is to have them go to the FDA early for pre-IND and de-risk the future IND and also possibly make their study better from an international perspective. However, the FDA, if the data are generated in accordance with good clinical practices and under an ethics committee, and the data are well presented and well reported, there's absolutely no prejudice from the FDA as to where the data are generated. Australia is considered an excellent region for clinical trial execution. So I would say there's no prejudice, but we do think that at least a pre-IND consultation followed by a CTN, followed by an IND, if not pre-IND consultation and a CTN under IND are preferable. And that's not to generate more business for GZP. It's simply, we found that programs are de-risked, plus having the FDA stamp of imprimatur on your program attracts more investment, we've found, because the FDA has reviewed your program, said what it said, and you're executing the program per FDA suggestions. The best timeline for the first pre-IND meeting, generally, give or take 6 months prior to your projected IND submission date. That's a good time, but it could be a year. We don't suggest much longer than that because sometimes science and medicine, particularly in rapidly developing fields such as oncology, change. And so you get the FDA's advice at the moment based on the data you've submitted and the program you've laid out. So generally 6 months plus before you want to submit the IND. It could be 3 months, but probably not more than a year would be a good idea. And you just have to allow the total execution time for a pre-IND meeting, which we typically cite as 75 plus days to generate the letter. Once the letter is received by the FDA has 21 days to accept or reject the meeting. If it's a good program, they'll generally accept the meeting. And then the meeting package needs to be submitted at least 30 days prior to the meeting. So if you get lucky and the FDA sets a meeting 60 days out from the time they receive the letter, then you might have to submit the package in a week because the letter might take 21 days to get to you. So essentially, when you submit the letter requesting the meeting, the package should be far along. The animal efficacy rule, is this translational oncology in pets? The latter part of that, I would say no. The animal efficacy rule is intended strictly for biological, nuclear, and chemical warfare agents, which either cure, mitigate, or prevent disease in those exposed to biological, nuclear, or chemical warfare agents. And the reason for it is because you can't ethically expose a human to those agents to test for efficacy. So what you do is you do safety testing in normal human volunteers. You do animal efficacy testing in an animal model or an animal that's exposed to a chemical, nuclear, or biological warfare agent. And you actually test efficacy extensively, preferably in at least 2 animal models. So you get animal efficacy and human safety bonded, and you can actually get approval for that product. Approval, or in the case of a biologic licensing. And it's not translational oncology in pets. There is another question. Confirm with an IND in musculoskeletal conditions, for example, one can add a clinical study for OA or tendinitis based on the submitted IND file. Well, I think both of those, I mean, OA has some autoimmune aspect to it. RA has a major one. I would say that if you have an IND musculoskeletal conditions, you probably can add a clinical study for OA or tendinitis to it if that was not the subject of the original IND. But if you cross reviewing divisions, you will have to submit a new IND. okay, track record of fast tracking from pets to humans. Hence, the interesting comparative oncology. Is the canine cancer data go into the submission or add to phase 2-3? The canine cancer for a human cancer indication is basically considered pharmacodynamic data. They are not clinical data because tumors in animals are not tumors in humans. So you can use the safety data from the animal to some extent, and certainly the efficacy data as support for your pharmacodynamic studies. There is a track record in some cases that's true, and oncology in some dog models is actually a good predictor of humans. So it's pharmacodynamic data, but they cannot substitute for human data per se, if that helps. How does the FDA view US companies come to Australia to do a CTN without an IND? Well, I guess that was asked and answered already. It's perfectly fine to do a CTN trial without an IND. There's no prejudice at all. But when you get to the IND, you might have some problems. I mean, our experience has been that CTN trials are often very well done in Australia as far as clinical is concerned. But if you haven't gone to a pre-IND or don't have an IND, the program is usually deficient to move ahead. For example, phase 1 or phase 1/2 under CTN, and you want to do a substantial phase 2 under IND. That often results in some deficiencies being noted during the IND review, could lead to a clinical hold, but it's very specific to the program itself. It's an excellent question. One more call, another 5 minutes or so. Okay. Okay, one more minute, and if we don't see any more questions, then we will close. And once again, appreciation to LSQ, to Sam South, to Lauren, and to all of the others who have contributed to this webinar. Question, is the FDA still well staffed under Trump? Yes, there are some disgruntled FDAers, but they're still there. COVID-19 has created a major firestorm, and quite a few FDA reviewers have, for periods of 2 weeks or longer, been delegated over to COVID. So we've seen some of the programs that we're maintaining something like, 25 programs for clients simultaneously at the agency. We've seen cases where a project manager has been temporarily reassigned, but the work continues. Sometimes the answers to less than urgent questions are taking a little bit more time, but in general, the FDA has been performing well. And yes, the FDA is still well staffed under Trump. I'm not sure if with the latest peccadillos of the present commissioner, what's going to happen, if frankly the interference with the regulatory and scientific processes continues, there might be some bad fallout, but we're certainly not seeing that now. It's a good question. Well, I want to also thank all of you for participating. This was one of the best question sessions we've ever had in one of these webinars. And it's stimulating and exciting to hear all of you working towards common goals. And may you all have a good and safe and prosperous and healthy last few months of the year. All right. Thank you all very, very much.